show you a number of pathways. I will never ask you to draw these. I'm using them to demonstrate some of the things that I've already talked about. So we've already talked about this is herpes simplex virus. It's a double-stranded DNA virus. So again, it has a capsid. Here are the spikes that it uses to engage. This works by fusion of the plasma membrane and the envelope. The DNA gets basically delivered into the cell, where it goes into the nucleus, where it's replicated and messenger RNA is made. Some of the messenger RNA goes out, and then of course proteins get made, and they have to come back because this is a virus that gets packaged in the nucleus. Okay, so the proteins have, obviously the messenger RNA has to leave, proteins are made in the cytoplasm, and then those proteins come back into the nucleus, and it's actually here that the spikes get put into the membrane, nuclear membrane, and the budding comes off the nuclear membrane, and then these would collect right under the plasma membrane, causing the plasma membrane to basically open and release them. So this is demonstrating some of the classic ways in which you can package a virus. And again, the early mRNAs are involved in synthesis of early proteins that would be needed for viral replication. So if there's a unique viral replication protein needed, then in fact the cell will have to support the virus by making it. Here we have HIV, which we've already been talking about, where I already showed you. It again fuses, and then the virion gets ejected basically into the cell, and then the virion or the coat, the capsid, breaks apart. We're left with the genetic material, which is RNA, and the little green dot is the reverse transcriptase that came with that RNA inside the virus that's packaged. That viral mRNA is reverse transcribed, and then the second strand DNA is made. The DNA gets injected into the nucleus. The provirus is made because that DNA gets injected into the host chromosome, and then it gets replicated, and then at the same time mRNA is made. So the mRNA is going to be needed for both the proteins, but also we have to make more RNA for packaging into the genome. So we have two different RNAs being made. Proteins get synthesized in the cytoplasm, and the gets packaged. And then note when the virus finishes off, you notice that all of the important spike proteins get embedded in the plasma membrane of the host cell. So this is the host membrane. This would become the membrane of the envelope. And basically there's a unique base placement of the spikes. Wherever the the virus is going to pinch off or bud out, that's where the spikes are placed. So that means that the host proteins that are present in the plasma membrane. So there'll be host proteins in the plasma membrane as well. So basically where you get budding off, all the host proteins will be excluded and viral proteins will be put into the plasma membrane. I'm going to show you that again in a little while. Okay, so again, three classic ways in which viruses get replicated. The double strand, in terms of RNA, we have a double-stranded RNA virus where we get an unwinding. You end up with a minus and a plus strand. The plus strand acts as a messenger RNA. The minus strand acts as transcription to make another strand, and then it gets assembled. We can also have classic positive and negative single-stranded RNA viruses. So again, remember the minus single-stranded RNA first has to be transcribed into plus strand, which is equivalent to mRNA. Transcription is by an RNA-dependent RNA transcriptase. Do we have such a protein in our bodies? All of ours are DNA-dependent RNA polymerases. So in fact, this protein has to be encoded by the genome and actually has to be carried in the viral particle. So then we make complementary that can both make proteins as well as making copies of the genome again for assembly. If it's a plus single-stranded RNA molecule, remember it can immediately be used to make proteins, but then this plus strand needs to be transcribed by a viral RNA polymerase, again, uniquely carried by the virus, that results in a complementary strand that then can be used to transcribe more single-stranded RNA. So those are the classic ways the three types of RNA molecules are processed. So let's spend a little bit of time talking about retroviral structure and the way in which they get packaged. So here again is our retrovirus. Here's the RNA. Here's the reverse transcriptase as a protein attached to that genome. Here's our viral capsid, and here's our envelope with the spikes. The genome has encoded into it there are more than 3 genes, but there are 3 important genes: the GAG, the pol, and the envelope. So the pol is the polymerase, the reverse transcriptase. ENV is the spikes that will end up in the envelope. And GIG, the GAG protein, is important also in the packaging process. So this is the pathway again. Remember, this is just to demonstrate the uniqueness of the retroviruses. Again, we get uncoding. We end up with reverse transcription. And then the second strand DNA synthesis. We now have a double-stranded DNA molecule that gets integrated into the host DNA. So we are now in the nucleus. That host DNA gets replicated, and so does the virus. We end up with viral, a transcription step here, back to single-stranded RNA, where it gets packaged into the nucleocapsid. And again, we are now back out of this nucleus, and it gets actually embedded in its envelope at the cytoplasmic membrane. So again, not all viruses that end up in the nucleus actually get packaged in the nucleus. So the retroviruses, even though their double-stranded DNA molecule, the intermediate, enters the nucleus and gets embedded into the host DNA, that single-stranded RNA comes back out of the nucleus and it gets packaged in the cytoplasm. So, viral glycoproteins, so this is the membrane. So this is a virus that's going to have an envelope. And this is an important thing for you to be able to visualize that I've already shown you in these various slides. Here we have a virus that's already enclosed in its capsid. And as it approaches the membrane, whether it's a plasma membrane or a nuclear membrane, what ends up happening is the spikes get embedded in that region. So remember, these spikes are viral glycoproteins. So again, you've got to imagine that you have a membrane on the host cell, and it has its various proteins. And what's going to happen is that this is the location where the virus is going to bud. We have all the spike proteins that have to be embedded in this membrane. But what also has to happen is these proteins in the host cell have to move away from this region, so that only viral proteins end up embedded in this membrane. If that process does not take place and the virus pinches off with both viral proteins and host proteins in the membrane, what do you think could happen to the host cell? Again, the envelope only contains viral proteins, so we have to exclude the host proteins that are in that region. What might happen if instead of getting a virus with only the viral proteins present, we actually end up with some host proteins embedded in the envelope of the virus?
Might affect the ability to adhere.
Okay, it could alter the ability to adhere, but it still has its spike, so it might still adhere correctly.
The host could die prematurely?
Sorry?
The host could die too soon?
The host could die too soon. I think you're on the right track. Basically, yep.
Would the immune system develop recognition of self-protein?
Correct. So now, when this gets presented to the host, and it's reacting to the viral proteins, it's also going to react to the host proteins with the possibility then of developing what? An autoimmune response. So it's actually critical that this packaging happens correctly, but sometimes a few host proteins may end up in this process. So it's really interesting that the process requires exclusion of the host proteins and the embedding of the viral proteins into the membrane, be it a plasma membrane or a nuclear membrane. So this is sort of showing you this again. Here we have the plasma membrane. We have hemagglutinin, which is kind of a baseball bat shape, and here we have neuraminidase, which is the lollipop. So hemagglutinin and neuraminidase are two viral spikes found on influenza. Here is our capsid, which has another protein surrounding it. And in green is host proteins that are also embedded in the membrane. So you notice as this virus approaches the membrane to be exocytosed, notice what happens to the green proteins. They're being excluded from that region such that when the virus pinches off, there is no host protein associated. The other problem that could happen is that viral proteins may end up staying with the host cell. Okay, so the other scenario is that viral proteins stay associated with the host cell. So again, we have a scenario where your immune system may start attacking your own host cells because of if there's some viral proteins left in the host cell. So in this diagram, you can see there are still some viral proteins. If that happens and the host cell has residual viral proteins, then in fact your immune system might start recognizing it as a viral particle. So the fact that the virus tends to do this fairly accurately is actually good for us, because otherwise the potential damage to the host cell would be even greater. So again, the best way to study viruses is by doing these plaque assays, where you determine the number of viruses in solution. And I don't want you to think that you can only do it affecting bacterial cells. You can infect animal cells with viruses and do the same kind of titer, where you look for clearing or partial clearing, which you can actually count. So, and you do the assay in a similar way, where you add the cells and the virus to a molten agar, you put it on top of the plate, and then you look for the presence of plaques. And plaques are only going to be produced when cells have been infected. Now, the reason you tend to do this most with bacteriophages is because they form what are called very clear plaques, because they're lytic. But if you're a virus that causes only partial damage to the host, you actually get a sort of an opaque appearance to the phage, because the virus is not completely lysed the eukaryotic cell. Okay, so let's turn our attention for a little while to influenza. So here again is an EM of influenza. Here's the structure of influenza with the RNA genome. It's in 8 segments. It has to encode an RNA, a viral RNA polymerase, as well as an RNA endonuclease, because remember, the genome of influenza is in 8 segments. So when it is first made, it is made large, and then it gets cut into segments. Neuraminidase is a sialidase, which means it removes sialic acids from glycoproteins. The theory of why neuraminidase is there is because it can remove these sialic acids from the rest of the receptors that are found on the host, and it may actually help to alter the mucus covering of the membrane. So remember, the host has glycoproteins. That's what the receptors are, and almost exclusively in the world of animals, the type of external glycosylation is called complex glycosylation, which has sialic acid residues as the last sugars. The hemagglutinin spikes, which are shown here, cause host cell membranes basically to bulge inward to allow for the receptor-mediated endocytosis. Lest you think there's only one major group of influenza, there's actually 3, influenza A, B, and C. It's influenza A that causes pandemics, which is epidemics that spread over large geographical areas, and they vary in both the H and the N. So their hemagglutinin and neuraminidase spikes are different. They can change. Influenza B is usually generalized localized outbreaks. There's no clear subtypes, so there's no difference between neuraminidase and hemagglutinin. Why am I telling you this? Because this is how we get strange virus infections in influenza A, the ability to mix up neuraminases and glutens. Influenza type C only causes mild symptoms and there's no clear subtypes. A and B can cause pandemics and epidemics almost every winter, but it's really influenza A that we have to be concerned about to resort and form these unique viruses. The word pandemic tends to scare people because people think, well, a pandemic is worse than an epidemic. And a pandemic, therefore, must make people sicker. Not true. A pandemic is just defining the geographical spread. So something that is pandemic means it's over a large geographical area, so it's crossed continents. It's making no statement about the severity of an infection. So when you hear there is a pandemic of influenza, it does not necessarily mean that influenza is particularly virulent. It just means it's managed to spread over multiple continents. So there are two important genetic variations that can occur in viruses, and influenza is probably the best one to look at these. One is antigenic shift, and the other is antigenic drift. So you can think of antigenic drift as a very slow change in genetic sequences. So basically this is the classic mutations that occur over the course of evolution over a long period of time. Antigenic shift, which is a term that's specifically used for influenza, in other viruses it's called viral shift or reassortment, really is about two different influenza strains coming together, resorting their genetic material such that the virus that comes out is completely different than either of the two that went in. So this is how we get a bird flu and a human flu going into a pig and getting H1N1, which led to the major epidemic we had a few years ago. So antigenic drift, which is a slow mutation rate, can occur in all the influenzas. Antigenic shift only in influenza A. So it's influenza A that we are most concerned about the outbreak of these very severe virulent influenzas. And really what it's about is modifications of those spikes, hemagglutinin and neuraminidase. So the viral pandemic of 1918, which was the first really major pandemic when the flu spread over many geographical areas. Why? What happened in 1918? Well, the end of the First World War was ending, people were returning home, and they all brought influenza back to their shores. And the type of influenza at that time was H1N1. So basically, H is hemagglutinin, N is neuraminidase. The number after it is the type subtype. So you can have H1N1, H1N9. So they're basically different subtypes of those two spikes. This was a particularly bad influenza because literally it caused people to drown as a result of the damage done to the lungs. Now, I've already defined antigenic drift as small changes, which really will not result in major types of modifications of influenza. But antigenic shift can lead a virus from going from H1N1 to a different virus completely. Okay, so antigenic shift then can occur when an individual is infected with a human type A and then is exposed to another virus. Okay, so if I have a virus, a particular type A virus, influenza type A, and then I get exposed to another virus, maybe it's an avian flu virus, and that flu virus enters my body, theoretically it of itself cannot infect me, but it can talk to the other influenza virus. And it can undergo antigenic shift to result in a reassortment of a virus that may well then be able to be spread person to person. So most of the bird flus that you hear about, fortunately for us, can't go person to person. So people who work with these birds get bird flu because they're exposed to the bird. But if you're exposed to the person who's gotten bird flu, you don't get flu. The problem becomes when the reassortment allows human-to-human transfer. So it's really a reassignment of the virus. So again, antigenic drift, if you pay attention to the triangles and the popsicles, popsicle being the N antigen and the triangle is the H antigen, what you see is there's slight variations. So there are one or two mutations that will change up hemoglobin a bit, or the neuraminidase a bit, but not significantly. But in the case of antigenic shift, what you have here is, here's virus A, here's virus B. Their genomes are different. Their neuraminidase and their hemagglutinin are different. They get together, they reassort. So here's two different versions of the reassortment. So their genome has fundamentally changed. So here we have added into this genome basically a brand new triangle. The triangle is the hemagglutinin. So now I've matched the neuraminidase of this virus and the hemoglobin of the other virus. I have a completely different virus now. Or down here, I've done a similar exchange. These now are highly problematic viruses because they're completely different. And they may have been a virus we've never seen before. And if it can go human to human, this antigenic shift that can take place can result in brand new viruses evolving. So, this could be in a scenario. So here we have a bird. He has a flu virus. Here we have a human. He has a flu virus. Well, pig gets infected with a human virus. By the way, pigs can support influenza, humans can support influenza, birds can support influenza. Then the pig gets infected with a bird virus. Those two viruses come together and talk to each other and they reassort, forming a brand new virus that now can move human to human. By the way, that's what happened in 2009 with the H1N1 virus. The reassortment occurred in a pig. A bird virus and a human virus talked to each other and we generated a new virus. So if we think about what viruses do, in their simplest form, they inhibit host DNA, RNA, and protein synthesis because they're hijacking the host's biology to make multiple copies of themselves. Many viruses are cytocidal, so the coronavirus, which is a naked RNA virus, the herpes virus, which is a DNA envelope virus, and the adenovirus, which is a naked virus as well. Cytocidal means they kill the virus, they kill the host cell. So they can kill, or they can simply damage. So for example, one of the steps I showed you when a virus enters is the formation of an endosome, which fuses with lysosomes to form a low pH compartment that allows the DNA to be uncoded, or the RNA to be uncoded. So cell lysosomes may be damaged, and if we damage lysosomes, what do lysosomes release? They're going to release those hydrolytic enzymes into the cytoplasm and cause damage to the host cell. Viral infection can alter the plasma membrane. So I gave you two examples. We could end up with viral proteins staying with the plasma membrane, or we could have plasma membrane proteins going with the viral particle. Either way, that can cause autoimmune responses to take place. Many of the proteins that are made in large amounts by viruses in of themselves within a cell can be toxic to a cell. Our cells don't like to make large amounts of protein. Our protein synthesis machinery is well controlled. So if we ask a eukaryotic cell to make a lot of a single protein, it usually gets sick very quickly. So therefore, when they're making large amounts of these viral proteins, those cells can get sick. What they'll often do is try to put those proteins into an inclusion body. So very often we see these dark, very dense structures in cells that disrupt cell structure and therefore, again, cell function. We've talked about the ability of some viruses to be able to integrate into the host chromosome and form a provirus, like the herpes viruses and the retroviruses. The adenoviruses can also do this. So the host cell may not be directly destroyed by the virus, so by cytocidal viruses. They may even not be fully damaged, but they also can be transformed, as we've talked about, into malignant cells. So cancer can occur with the retroviruses, the papillomaviruses, the herpes viridae viruses, and examples of the herpes viridae that can do this are Epstein-Barr, the herpes virus, and the hepatovirus, which is for, the Epstein-Barr and the herpes virus. The hepatovirus can also do this. This is actually an RNA virus. And also the hepatnoviridae, which is a DNA virus. Does anybody know what Epstein-Barr can cause besides cancer? It causes Burkitt's lymphoma. But more simply, that tends to be a viral infection that young people often get. Mononucleosis is caused by Epstein-Barr. Epstein-Barr is also thought to be the cause of the nondescript disease infection that causes severe exhaustion. And although that's a little more iffy as to if it is the cause. All right, so any virus that can alter genetic material in the host can cause transformation. Okay, types of infection. We talked mostly about acute infections. They cause damage to cells in very short time. But there are also the persistent that last years, either as a chronic viral infection or a latent viral infection. So herpes B or HIV is an example of a chronic viral infection. Symptoms can be very mild or absent for long periods of time, and then the person presents with the symptoms. Latent viral infections are when the virus completely stops reproducing and remains dormant. During latency, there won't be any symptoms, and basically, nor will there be any antibodies or virus detected. So the classic example we'll use is the herpes viridae, which is the herpes simplex virus. It can cause cold sores. Herpes simplex virus type 1 causes cold sores. Type 2 causes genital herpes. This is a contagious disease, by the way. So if you come in contact with somebody, even a blast from somebody who has a cold sore, then that you can actually get the virus from them. Cold sores are not canker sores. Often people mistake them. They're blisters. They're actually blister that's full of viruses. Generally, the first time you have a herpes simplex virus, it doesn't manifest as a cold sore. It'll manifest like a flu-like symptom where you feel sluggish, you just feel awful, you have swollen glands for a week or so, and then subsequent outbreaks will occur. Some people get them 4 to 5 times a year, triggered by stress, illness, or fatigue. Epstein-Barr, which causes mononucleosis or chronic fatigue syndrome. Every one of us probably, if we tested, you would have the Epstein-Barr virus present. You've been exposed to it, it did absolutely nothing to you. It didn't cause mononucleosis or chronic fatigue symptoms. So for most people, they're asymptomatic, but it can lead to autoimmune diseases and Burkitt's lymphoma. And of course, another good example is varicella zoster, which is chickenpox, which is a herpes virus as well. It's the acute infection. Shingles is the latent infection. And most people would argue that the more severe infection is the latent infection, which is shingles. For anybody who suffered from shingles, the suffering is far more serious than the original infection. So how does chickenpox as acute and shingles latent result? Shingles, by the way, is also called herpes zoster. So that for every 1 in 10 people who have had chickenpox as a child, they'll get shingles as an adult. Usually in their 60s and 70s, although there are examples of children that actually manifest through shingles. It's all about your antibody response. So if you get chickenpox and you don't get a high antibody titer, and then the virus hides and reemerges, you're more likely to have shingles at a younger age. Do not think because you have had the chickenpox vaccination that you cannot get shingles, because again, if your antibody response is low and the virus is hiding, yes, you can get shingles, even if you have the chickenpox vaccination.